Hopefully you can see my slides. Is that right?
Yeah.
Can somebody confirm that the slides are up?
Yes, yes, we can see you.
Great.
We can see.
Okay, thanks. Anyway, I'm really, really sorry not to be there. I would have loved to be there in person. I enjoyed the last two talks, and I'm sorry those were the only ones I saw because it's the crack of dawn here on the west coast of America. And I'm really sorry to miss Joachim, who's a very old friend. So I hope the meeting has been really lovely and fascinating. The last two talks were great. So as Trevor said, I have recently turned my attention to cryo electron tomography. And this work is the result of support from the Chan-Zuckerberg Initiative. And the mission of the Chan-Zuckerberg Initiative is to make it possible to cure, prevent, or manage all diseases by the end of the 21st century. People always say, oh, that's very ambitious. I personally think that is not beyond feasibility when you think where we were a century ago and how far we've come. But what's slightly unique about the Chan-Zuckerberg Initiative is they support this work in a myriad of ways, but in particular, they give support for basic science and technology and support to develop new technologies. And that's a little bit unusual in the world today. Anyway, as a result of that support, they decided to establish the so-called Chan-Zuckerberg Imaging Institute. It was established in 2023, sometime last year. And the mission of that institute is to enable insights into the architecture of complex biological systems at the molecular level, basically to understand how cells work, how they interact, what their dynamics are. And the idea with the Imaging Institute is going to have a number of grand challenges. And the very first one is to understand the molecular architecture of the cell to near atomic resolution. That's basically cryo-ET, as I'll describe. So your question might be, well, why do we need to understand cells at atomic resolution? And that's because cells are very complex, very crowded. They're filled with dynamic molecular machines. And the function of these machines depends on local context. They're not just on how they behave in isolation. So understanding these cells was thought to be fundamental to the overall mission of the CZI to deal with disease by the end of the century. So then the next question is, how do we understand cells at atomic resolution? Well, you've been hearing a lot about cryo-electron microscopy. That's one of the structural biology methods, along with X-ray and NMR, that really resulted in tens of thousands of solved structures as a result of really 40 years of very hard work by structural biologists, at the end of which AlphaFold, the machine learning technology, came along and was able to predict millions and millions and millions more based on sequence. So it was this fundamental knowledge that went into public databases that led to this explosion now of knowledge about all kinds of structures. But wanting to know how a cell works by looking at its isolated components is like wanting to know how does a watch work by looking at all the pieces of a watch that are sprayed out onto the table. We obviously need to understand how things are in context. And right now, the only game in town for that is cryo electron tomography. If we want to know at high resolution what is happening inside the cell, that is the method we must use. So a quick primer on cryo-ETs will only take a few minutes. So if objects of interest are small enough, they can be vitrified like single particles are vitrified. You can put them on a grid, reduce to a thin film, plunge, freeze, and then you get one of these entities. You then can put that inside a transmission electron microscope. The electrons go all the way through this object, and we take a series of snapshots. We tilt the image and take a snapshot, tilt the image, take a snapshot, and we create what's called a tilt series of images. We can use that series of projection images to reconstruct a 3D volume of the cell that we were looking at. And that's completely analogous to going inside a CT machine, an MRI machine, and getting cross sections of your brain without actually having to slice your head open. So that's the analogy for the two. Once we have these 3D volumes, we can see things like ribosomes in there. I've circled a few of them there. And if we can gather enough of them together and average and align, and we can do a reconstruction analogous to how single particle electron microscopy is done. However, if your cells are very large, and most mammalian cells are, you need to use a special device called a high-pressure freezer to get your cells well vitrified. At the end of that, you have a big clump of cells. And one question you might ask is, well, where in this clump of cells do I go look to see what I want to see? And we use a method then called cryo-confocal light microscopy. Basically, light microscopy, confocal light microscopy, but with a cryo stage attached, so you can keep this sample frozen. Now I might see, these green things here, that's what I want to look at. But this clump of cells is way, way too thick to put into a transmission electron microscope. You need to get the electrons all the way through. Excuse me. So we use yet another device called a focused ion beam scanning electron microscope. And this is a scanning electron microscope with a focused ion beam, which is basically a knife that chisels away parts of the cell. And what it does is cut away most of the cell and leave a little thin window into the cell that's left behind. And if we look at what that thin window looks like in the scanning electron microscope, here's an example of what we call a lamella. This window is often referred to as a lamella, and these are yeast cells. These whole things here are yeast cells embedded in that thin window. Now it's thin enough, we can stick it back in the transmission electron microscope, and we play the same trick of tilting, snapping, and producing our tilt series, and then from that, producing a 3D volume, slice right the way through. Now we can see this sort of beautiful myriad of structures. Here's a lovely mitochondria. Here's the wall of the nucleus. And if we go and look at a single slice, here we go. There's a nuclear pore. Okay, that's the nuclear membrane, and that's a nuclear pore. And this is maybe some microtubules. Here's a nice ribosome. Ribosome's ubiquitous, of course. And here's a ribosome inside a mitochondria. And again, if we can get enough of these ribosomes or whatever other structure we're looking at, and combine and average them, we can go back to getting a nice 3D structure at molecular resolution, near atomic resolution, at least interpretable resolution. But the problem with that is we need tens of thousands of these to average together to get to high resolution. So we need hundreds and hundreds of these tomograms. They're very noisy. And then we need to find tens of thousands of these particles in them to get to a high resolution structure. So the grand challenge, the first grand challenge we've set ourselves at the Imaging Institute is to try and sort out this cryo-ET pipeline. It's very complex. It has major challenges and bottlenecks at every step. We need all kinds of things. We need to label structures of interest. We need super resolution light microscopy, because it's really hard to find these thin lamella using the standard light microscopy. We need to improve our FIB milling. We need to improve the throughput of our cryo-ET. And then there's a very, very complex processing pipeline that we need to go through that has major challenges. So our goals are to develop robust, reproducible, openly accessible tools. In other words, to democratize this method, to enable it to take place at scale, and most importantly, to apply that to important key biological problems. You can't develop technology in isolation. You end up with something nobody wants. So we have to tie this very, very closely to the biological driver projects that need this method. So to serve that mission, we have a roadmap. And just to sort of generally, describe what the roadmap is, you first need a whole lot of technology, okay? Really expensive technology, big microscopes, FIB milling, tons and tons of computational resources. Then we have these technology drivers, and I'll talk about a few of those in a bit, and tightly linked, technology development projects, and then tightly linked to biological driver projects that keep us honest and keep this technology cycle virtuous. So that's what we did. This is where we are. We moved into this building last November. It's right about here. That's San Francisco. So we're just about 20 minutes down the peninsula from San Francisco. We hired a fantastic team of people. We'll be about 40 people by the end of the year. This all started middle of last year, so it's been quite a fast hiring schedule. And then we filled the building. Whoops, sorry, I didn't mean this to be making noise. We filled the building with all kinds of beautiful equipment. This is our first Krios being built. We got that delivered on October 11th, and we signed off 6 weeks later in December in the new building. So we have 2 of these big Krios machines. I'll describe the second one in a minute. We have a FIB milling device. That's an experimental FIB miller. And then we have a high throughput FIB milling device. This is a new instrument that's been made available from Thermo Fisher. And we have a cryo light microscope and a high pressure freezer and a bunch of other stuff. And of course, cell and tissue culture and all the rest of it that you need. So then let's talk a little bit about some of the technology research and development projects that we're involved in to try and democratize this pipeline, or at least improve it, so it can be much better and quicker. So one of the first is a very large project called the laser faceplate, and then I'll go on to describe a bit of the data processing, but we're working on some of these other things as well. So the laser faceplate, what I'm calling a dual laser faceplate, because that's what we're building. So this is this astonishing device developed by Holger Mueller and Bob Glaser at University of Berkeley, UC Berkeley. And what it is the ability to make an optical cavity that can make an incredibly powerful continuous laser beam through which the electron beam passes, and as a result undergoes a phase shift. You can have a whole lecture on just the faceplate, so I won't say much more than that, except that you need an extremely high power laser, 400 gigawatts per centimeter squared. Just if you don't know what that means, it's the highest power continuous laser on the planet, is in the middle of this microscope, and that gets you a phase shift of 90 degrees. And the way you do that is build this cavity, so you resonate the laser beam back and forth. You need near perfect mirrors, atomically flat mirrors at either end with 99.9% reflectivity. So it's a very technical device, but it works. And Holger showed this some time ago. I think, where was this? 2021. This is, a thin specimen, so just S20 proteasome, but even so, you can see with the laser on, you get this huge boost in contrast. And even more excitingly, if you have a thick specimen, this was sort of a manufactured thick specimen, just a thick layer of Rubisco. Even so, with the laser on, you can start to see the shape and size of these particles. And this is more analogous to what we want to do inside a cell. This would be, more what the crowded environment of a cell looks like. So we're very excited about that. And the problem is, this is what Holger's lab looks like at the University of California. It's only a situation that a laser optics physicist could love. And indeed, it takes 2 or 3 people to run this when it's running. But so our job at the Imaging Institute is to take that and miniaturize it and jam it all inside a normal Krios and make it very easy to use, have kind of a basic on-off button. This is an incredibly complex project, and it's been a collaboration between us and Chan Zuckerberg Institute, University of Berkeley, Holger's lab, Thermo Fisher, who designs the microscope for us, and an engineering company called Applied Precision Design. So the first step was to design a microscope that would accommodate this dual laser faceplate. I'll tell you in a minute, we're building 2 of them, not 1, and give you decent optics on the column. So Thermo Fisher did that for us. They designed a new column that would accommodate the laser faceplate and gives quite good optical readouts. And the most amazing thing about this design is it sped up the delivery of our instrument by a year, and that never happens. Usually it delays everything by a year, but in fact, this sped it up. And indeed, that's the instrument that got delivered about 4 weeks ago, I guess. It's all assembled now. It's huge. It's very tall because there's a new piece of the lens in here. And this port here is where the optical cavity will be inserted. This is where the specimen is inserted, which is showing you how tall it is, because that's Angie from our team measuring how to get a sample in. And all this space here is available for these optics boards, input and output optics boards that will support this dual cavity. So it's a dual cavity. There's 2 mirrors in this direction, 2 mirrors in this direction. This is the insertion arm to get it in there, and this is one of those input optic boards that sits about here, and the others are tucked here, there, and everywhere into the column. Okay, so sorry, that's basically what I want to say about the laser faceplate. I guess I'll just move on, Trevor, and you'll ask questions at the end if there are any. So moving on to a completely different topic, that's hardware, and we're doing a lot of biochemistry, hardware, etc. But of course, a lot of the cryo electron tomography pipeline is software. When we're at the microscope, we end up with these jiggling, jaggling bag of jelly. That's the raw unaligned tilts. And what we have to end up is a beautifully aligned series of slices. And from that, or from this original set of tilts, we need to extract high resolution structures. Only one has really been done. The ribosome has been done. These I'm just showing as cartoon form because we haven't really gotten, maybe there's a few exceptions, but not many exceptions of getting high resolution information out of these very noisy images. The pipeline, the processing pipeline is also horrendously complex. If you ever look at, one of the, hello, sorry. Oh, sorry, I think somebody's talking, but let me know if you want me to stop. If you've looked at a methods paper in FIB-SEM, hello, I'm getting feedback and somebody's talking, so sorry. Let me know if you want me to stop, Trevor. Okay, I'm just going to plow on then. Okay, so long, complicated, sorry, I'm not sure where I am. Long, complicated processing pipeline. And what we want to do at the Imaging Institute is simplify that. So we really think of it in 4 steps. We need to do a bunch of pre-processing, which is turning that jiggling bag of tilty frames into a nice, clean, sharp 3D volume that's corrected in many ways. Then we need to annotate that volume. We need to find out where our particles are. Then we need to do subthermal reconstruction to grab the particles out, do the alignment and processing, and then, of course, interpretation. So I'll just talk about a couple of those today. And all along this way, we need to keep ourselves honest by measuring metrics to see, are we improving this pipeline as we go along? So the first step we've done is that pre-processing, and we've turned that into one-stop shopping. We go from the jiggly tilt series all the way into a nice, well-aligned tomogram that's corrected for motion, that's corrected for CTF, that's corrected for tilt. And we get a beautiful tomogram and a denoised tomogram. And we do that in one step, and we do it in real time. And by real time, what I really mean is as fast as we can acquire data. To me, that's real time. And you can do it that fast, then you don't need to go faster really, because you're not slowing down the microscope. So this software that is released now. It's called rTomo3. It's in the public domain. It used to be that it was not public domain, but CZII bought back the rights and put it in the public domain. It improves throughput, contrast, and resolution because it improves all these alignments. And just to give you an idea of the importance of this denoising step, this is a machine learning method to denoise. That's what a raw tomogram slice looks like, and that's what a denoised raw tomogram slice looks like. And you can see in the denoised image, I start to see the shape and structure of some of these very complex membrane proteins that are sticking out of these membranes. So it's a terribly important step. And we hope, of course, that the laser faceplate will give us some of that for free. Okay, so that's the pre-processing step. That's one well under control, and some of these other steps are also being looked at. But the huge, huge bottleneck right now is this annotation. It takes us real time to do this. We can get, hundreds and hundreds of tomograms in a few days. With enough compute power, we can do this in a few days, but this often takes many months or years. And as a result of that, very few things have been annotated. Ribosomes and membranes, great. Everything else, super difficult to do. So our model of a cell is like this instead of this lovely complex environment. So why is annotation so hard? Well, particle locations, we need tens of thousands of particles to get to atomic resolution. Cells are horribly crowded. Tomograms have horrible contrast. They're anisotropic. Individuals' particles are sparse. And the methods to do this annotation, to find these particles, are basically manual at the moment, or maybe template matching. But we really think the real hope is machine learning. If people can do it, we can teach an algorithm to do it. So annotation takes forever, and basically you sit there and pick these out, or maybe ribosomes, you can get some methods to do it for you. So to address this problem, we have started a cryo-ET portal, a website to provide annotated tomograms to the community so we can encourage people like machine learning developers to develop new algorithms for us to find these particles. And if you can go to that portal, there's a QR code if you want it. Otherwise, just type cryo-ET data portal into the web browser. You can go there and you can start browsing this data. So there's 15,000, well, maybe a bit more than that now, because we're adding continuously. You can go and look at these tomograms. You can search on various things like M. tuberculosis and find a tomogram and then look at it and look at the annotations of it and explore it in various ways, etc., etc. So what can we do with such a portal? once we have these tomograms, this was done when we had about 13,000 in there, that's improving. And then we ran a machine learning-based membrane finder across every tomogram in the portal. We could do this because everything is well annotated and the format is well under control. We could do this in a couple of days. We annotated all 13,000 of these membranes. And a different annotation software for membranes called TARDIS from Tristan Bepper's group has now done the same thing. So now we have these membranes annotated by 2 different methods, maybe more by now, and we can compare the methods and actually start to learn how they do and make them better. There's all kinds of ways of viewing the data on the portal. There's this interactive web viewer, which you saw briefly before, but it's got all these nice annotations now. You can use Napari, which a lot of the community uses, or there's a Python API if you're a code developer, want to just drag all of them over and start doing things with them. The next step for the portal is that we're running a machine learning challenge. Note that there's a $50,000 prize for the winners of this challenge. And what we're going to do is provide well-annotated ground truth data on some tomograms, and then ask the community to develop algorithms to be able to, do this automatically on tomograms that are sequestered away and are secure. And that challenge will be announced sometime this fall. You're all encouraged to enter it if you have new algorithms that you'd like to try on this data. I just want to mention, I don't have time for this because I think we're nearly out of time, but we do keep ourselves honest with really good biological driving projects inside the Institute. And so far, we're looking at organelles. We have a really fancy method of concentrating organelles down onto a grid surface. And then we can label, structures of interest on the surface of those organelles and go find them and do subtomer averaging of them. And this is a nice way to do a high throughput structure as we learn how to do these things and as we learn how to get the data better, so we need fewer particles. We're also looking at cilia and the ciliary synapse. So there's a synapse between axons and cilia that's discovered by Xushen Xu, who joined our group. And then we have a more sort of general project to look at synapses in general that are collaborative projects with, a half a dozen people in the community. Okay, with that, I think I'm done. This is the fantastic team that we have. It's an honor and a pleasure to work with them. And we're having enormous amounts of fun tackling this project. Thanks. Done.
Thank you very much, Bridget. Don't take your picture away. Hang on. Can you stop sharing and then we can...
I did.
Oh, you did. And why have we gone back to the IDM slide?
I don't know that. I can see just people on the screen.
Okay, so can you put her picture back on? Okay, they'll put your picture back on in 1 second. Okay, is everybody busy preparing their questions? Okay, Joachim wants to ask a question.
Hi,
I'm not asking a question. I want to say hello. And really, unbelievable, fantastic talk. And thanks very much for giving us the update. And they hired the right person. Okay.
Thank you so much. That's very kind of you. I wish I was there having fun.
We wish you were here too, Bridget. You must come and visit us. Okay, so I mean, what you've shown us is an absolutely amazing technological powerhouse, and it's really pushing the limit of what everybody is doing in the world, and it's showing us how actually this kind of science is increasingly beyond us unless we work with you. And we can clearly get involved at various different levels by looking at tomograms and the sections that you've made. So, but I don't think that anyway, let other people ask.
I just want to comment on that. I mean, I 100% agree that the technology you need is so sophisticated and expensive. But I do think, when I was at the New York Structural Biology Center, we started national centers. And I think Jason works at a national center in Europe. And I think there's more and more of a push to make a few consolidated national centers to provide the very expensive technology that people can come, and if we can get the throughput high enough, which we did in single particle, 10,000 images a day now, then, you know, those centers can cope with quite a high volume of work. And I think the same thing will inevitably happen for cryo-electron tomography, that the very expensive equipment will be at a few centers, people will send samples or come and use it, and then they'll go home for the much longer process of analysis, which can happen in other places as well.
Okay, Jeremy. Yeah.
Hi, Bridget. That was a great talk. I've got a question that I've asked actually like a few times, and I've never really got an answer that I kind of accept.
Uh-oh.
Okay. So this is the question. So you have this sample, which is relatively thin sample. You tilt it and you then get a 3D volume. Okay. You then pick out individual particles and you average them together, and you get a nice high resolution and relatively noise-free three-dimensional reconstruction. But I've noticed that often when you look at single images, it's the same sample. So in other words, if you, instead of sharing the dose throughout all of the tilts, you just use your entire dose for a single image, you can often see the particles in those single images. And I've always thought, well, can't you then just do a single particle type reconstruction? So in other words, you can pick those particles which lie in random orientations, and even if they're kind of crowded, you can kind of pick them out and do a 3D classification and things like that. Does it improve things to first do the tomography, or can you get the same results? Has anybody ever tried that? Basically.
Yes, in fact, there's a huge amount of effort on this. Nico Grigoriev started it in his lab, and some others at Geneva, and now Bronwyn Lucas at Berkeley is continuing. In fact, Bronwyn's 3 students just spent the summer with us because she was on maternity leave. So I know all about this. And basically that's what they do. They put all their dose into that one zero tilt image, and then they go using cross correlation to find particles in there. The problem right now is in the crowded environment of the cell, the only thing they can find reliably is a ribosome because it's nice and big.
So
anything else is very, very hard. We have high hopes that the laser phase plate might help that because you'll get better contrast, improve your signal to noise. And so you might be able to find smaller things. I think many people now believe, all the subtomo averaging is not done from the 3D volume, it's done from the tilts. You use the 3D volume to find where in the tilt to go to pick out your particle, because it's very hard to find those. Hence, Bronwyn's team can only find ribosomes. So, but you'll use a combination, and we're exploring right now how to spread the dose. Just use most of it in zero and then take the rest of it for tilting a bit, so you can kind of more or less locate your particles and go back to that zero tilt to find them, or exactly, how do you spread this dose around 2D to 3D? But absolutely, lots and lots of people are working on exactly that, and it does work very, very well for big things. Does that answer your question finally?
Yes, finally.
Okay. So you've taken away his picture again. Okay. Any more questions? Comments?
How to access the sensors?
Oh. Can we access your facility?
Not at this time. Our mission is to, well, if you collaborated, you could. It depends if you had a collaboration, and we're certainly open to collaboration. We're not a service center, though. Our mission is to try and improve the throughput and the access and the reliability and the robustness so people can start centers and start having people in. Right now, this is not like single particle at all. It's hard, it's long, it's slow, it's a slog. It's not for the faint-hearted. Cryo-EM, single particle cryo-EM, you have a really good sample in a test tube, you walk into one of those centers, you are going to get an atomic resolution map, in a day or two. So it's night and day different, the cryo-EM and cryo-ET centers. But I have high hopes that within a few years, don't know how many few is, that we will see centers like this where anybody can go and access. So get ready to access them. But right now, they don't exist, in that sort of throughput and volume that you can just take anyone in the world in.
Was that a question?